Okay, so let's talk about the immune system. So I know several of you took immunology already. So for you, this would be kind of a quick review. But I think it's very important to go through some of the main concepts of how our immune system works, if we are going to have a chance to actually understand the relationship between cancer and our immune system. So as an overview, our immune system has two kind of really important concerns with it. One is that it needs to have certain specificity. In other words, it needs to be able to recognize specific things and see those things in a background of otherwise maybe non-important things. So in addition to that, our immune system needs to also be able to adapt or develop memory against certain types of signals that maybe come from pathogens, things like that, so that our immune system is ready to deal with them if they see them again in the future. Another very important concern of our immune system, one that we often don't spend too much time thinking about when we think about what our immune system normally does, is this very important ability to distinguish between self signals and non-self signals. Now, what do we mean by that? So our immune system, as we will discuss, is very concerned with being able to detect certain molecular signatures that are present inside of our body. Some of these may be coming from different types of bacteria, pathogens, bacteria, viruses, other types of pathogens, right? And those types of molecular signals would be considered non-self in origin. But really, for most of the time, you are not undergoing some sort of infection or you're not sick. And so your immune system is constantly dealing with your self signals. In other words, all the normal healthy cells of your body presenting molecular signatures as well. And so the immune system has to tell the difference between the two. There are three main functions of our immune system. Very importantly, number one is it needs to be able to recognize and remove abnormal self cells. So these are cells that maybe have become damaged or are simply too old, so they've already served their purpose, but they're still kind of floating around in our body. They need to be identified, targeted by our immune system so that they can be recycled before these abnormal cells turn into something worse, like disease. So the immune system also has to be able to remove dead or damaged cells. And of course, our immune system is concerned with protecting against invaders like pathogens. But I put pathogens and invaders as number three, because that's usually the thing that we think about when you start talking about, oh, immune system, right? I think the first thing that comes to mind is your body protecting itself against illnesses caused by bacteria, viruses, parasites, whatever, right? And I think the average person on the street, if you ask them what your immune system does, that's what they would think about. But in reality, as I mentioned a few minutes ago, every single day of our lives, our immune system is preoccupied with being able to maintain the normal health of our body's tissues by recognizing and removing abnormal cells. So, of course, when we say abnormal cells, this ties in with our discussion of cancer, because I can't think of a more abnormal cell than a cancer cell. Clearly, then, in order for cancer to get as far as turning into an aggressive metastatic tumor, the cancer along the way must have found different types of mechanisms for evading or avoiding detection by the immune system. So a big part of our discussion today and tomorrow then is, how does cancer trick the immune system so that your immune system cannot do its job and eliminate disease? So to start off with, let's quickly look at some examples of cells in our immune system. So this table is from a human physiology textbook that I use for one of my other classes. So it's not in our cancer textbook. But on the top here are cartoon representations of 6 different types of leukocytes, or white blood cells. Of these 6, 5 of them are types that you'd find floating around in your blood. So these include things like lymphocytes, monocytes, neutrophils, eosinophils, basophils, and mast cells. So these over here on the left are considered granulocytes. You can tell just by the cartoon, they're filled with these little granules, right? So these are vesicles that contain different types of cytokines. So in other words, different types of signaling molecules, as well as other types of molecules that are kind of effector molecules, things like histamines or other types of inflammatory signals meant to help fight against something like a pathogen in the body. Monocytes circulate in the blood, and then when they exit the blood and they undergo diapedesis, remember that's the term that we give to cells that go through the blood and then exit out of a capillary into surrounding tissue, monocytes may differentiate and turn into macrophages that then crawl around in the tissue, kind of cleaning up. Macrophages are sort of like the custodians of our tissue. They go around cleaning up all kinds of scraps of leftover parts of cells that have undergone apoptosis, or dead cells that have died some other way. And of course, macrophages also go around trying to eat up things like bacteria and virus particles. Monocytes can also differentiate into dendritic cells. So dendritic cells you would not find in your blood at all. Right? So dendritic cells are only found in tissues, solid tissues, or in your lymph fluid. And dendritic cells go around kind of collecting all kinds of examples of different bits and pieces of things that they find in the tissue. And then their job then is to carry these around and present these different types of molecular signals to other immune cells, so that those other immune cells have an opportunity to be alerted or aware and activated against these signals, even though those other immune cells might not have been in the original tissue where the dendritic cells found these signals. All right. So this table here, it's not something for you to memorize, but it's a very nice kind of layout for understanding how these different types of cells are categorized. So as I mentioned before, this group of cells over here are the granulocytes, because they contain these vesicles that can release different types of substances when the cell is activated. These cells, so there's some overlap here, right, are phagocytes. So that means they actually deal with eating other cells, small cells like bacteria, viruses, etc. These cells over here are cytotoxic, meaning that their job is to release different types of signals that cause a target cell that they are joined up with to go ahead and actually undergo apoptosis. So certain lymphocytes are T cells, certain lymphocytes are B cells. And the B cell types of lymphocytes can turn into plasma cells to release antibodies. So these lymphocytes are considered cytotoxic. Eosinophils, there's some functions of them that can be considered cytotoxic as well. If you look at the relative percentage of these different types of leukocytes in your blood, you can see that some are very common. For example, the most common ones that you'd find floating around in your blood are the neutrophils, followed by things like lymphocytes. And this would go down to the level of very rare white blood cells, things like eosinophils and basophils. This row over here just gives you some other common names that these cells are often referred to as. So I think this is kind of useful because it may be that you've actually heard of these cells using one of these other names here. And this last row is very useful for just getting an understanding, sort of a quick overview of what these cells, how they function together. Okay. So let's talk about what happens when we actually become infected by something like a virus or bacteria. All right? So we can use this as a model first for understanding how different parts of our immune system work when dealing with a foreign invader. And then that makes it easier for us to understand when we shift our discussion to how our immune system deals with internal threats coming from things like cancer. All right? So this cartoon has us look at something like if, let's say, a bacteria has invaded our body, and this is a bacteria that maybe your immune system has not seen before. So therefore, your immune system is not really aware the best way to sort of deal with it. Here we have some B cells. So this is a naive B cell, a type of lymphocyte. And this B cell on its surface has a unique receptor, B cell receptor, which is a protein that we call an immunoglobulin. So these immunoglobulins are expressed on the outside of this naive B cell, and only this B cell would have the particular specific shaped immunoglobulin that's being carried here, because this is how it developed. The B cell developed in the bone marrow to come up with this unique three-dimensional protein that can bind antigenic signals. So an antigen is some sort of three-dimensional molecule that can be received by receptors in our immune system and actually used to go ahead and stimulate the production of antibodies, right? So that's why the name antigen can be broken down into antibody generating signal. So in this case, this particular B cell binds to this antigen on the surface of this bacteria. So this stimulates this B cell to replicate and make many copies of itself. Because as I mentioned before, this B cell at the start was very rare. It is unique. But now that its immunoglobulin on its surface has been identified as being something potentially useful and valuable, the B cell wants to make many exact copies of itself so that there are many more instances of B cells that have this exact immunoglobulin on its surface. Some of these B cells, these identical clones, are then stimulated further to differentiate and turn from a B cell into a plasma cell. That plasma cell's job then is to go ahead and release a soluble or free-floating version of that immunoglobulin that it has on its surface, and that free-floating immunoglobulin is what we call an antibody. The idea here then is now you can actually flood your circulation with billions and billions of these antibodies, all of which then, as they're flowing around in your body, if they encounter the bacteria, right, can go ahead and bind to that bacteria and help facilitate your immune system's ability to fight against it. This little chart down here gives you an idea of your antibody titer, or levels of specific antibody, following infection, if indeed this is the first time that you've been exposed to this bacteria. So in other words, this is your primary immune response, right? The first time your immune system has had a chance to develop a defense against this specific bacteria. And you can see over the course of 1 to 2 weeks, the levels of these high quality antibodies peak in your circulatory system, right? So we're talking about these little green antibodies here. The levels peak. Importantly, the actual time in which you feel sick might be only, let's say, the first week, week and a half. And if this is your kind of typical run-of-the-mill cold or flu, something like that, I think we all realize that after a week, week and a half, we're sort of back to normal, right? If we have a healthy, robust immune system. But our immune system in the background is still making tons and tons of high-quality antibody, so that when we return to school or work or wherever it was that we were exposed to this pathogen in the first place, we can certainly expect that just because we're back in the classroom now and we're healthy, it doesn't mean that our classmates around us are all healthy and over it also, because everybody was probably taking turns getting sick. So you will be exposed again to the same pathogen. So this increased level, this sustained increased level of antibody in your blood for the next few weeks will help protect you every time you breathe in that same virus particle again, or be exposed to that bacteria again. So up here with the plasma cells making antibodies, this is a key feature of your primary immune response. Some of these B cell clones that were generated over here are not told to turn into plasma cells. They are encouraged to stick around. So after the infection is beaten and you are free of the virus, and you no longer need to have really high levels of antibody circulating in your system, because maybe for months or years later, you're not exposed to this virus again. Right? You want to have some memory of what your body came up with that was successful in fighting this the first time, so that you don't have to go through the whole process of getting sick and figuring it out again, right? So some of these B cells stick around as memory cells. So again, they have the specific immunoglobulin on their surface that was shown to be very effective in fighting this virus, let's say. And so you don't want to lose these cells. So on this cartoon, this memory cell kind of goes off the bottom of the cartoon. The next slide that we'll move to has this memory cell starting from the top, okay? So here it is. The memory cells are long-lived, and they continue to sort of divide every so often so that they are maintained in your body potentially for the rest of your life, right? That's how it is that certain types of diseases, you get sick from them once, you survive, and then potentially for the rest of your life, you won't get sick from that same pathogen again, right? Because you have these memory cells sticking around. So in our secondary immune response, this means the second time or any subsequent time in your life that you're exposed to that same bacteria again. So here's that orange bacteria again, right? This might be months, it might be years later, because you have these memory cells in a higher population in your body than when you first started, right? There's a much greater chance that some of these high quality memory cells will bump into the pathogen right away. And these memory cells will be much faster and quicker to respond. They will multiply right away, turn into plasma cells again, and make that very important antibody. So here is your secondary immune response. From the time that you are infected again, you can see your antibody concentration just goes shooting through the roof very quickly, right? Compared to the lag of 1 to 2 weeks experienced the first time you were exposed to this thing, right? But now that you have memory and you have these memory B cells, tons of high quality antibody are released, so much so that this bacteria or this virus never has an opportunity to even make you sick a second time. They are wiped out so quickly. Okay, so this then brings us to a discussion about how things like these B cells that have these immunoglobulins on their surface, right, how do they actually know what to do, and how do they coordinate with other immune cells in order to give you the appropriate type of immune defense or immune response? So let's start off on the left side of this figure. So here we have the virus that's invading the host here. So these little red things are the virus particles, little capsules. When they go ahead and infect a host cell, right, they hijack the cell and force the cell or trick the cell into making many, many more copies of the virus particle, releasing many more copies of the virus particle than actually originally entered the cell. And in the process, the cell itself might be destroyed. Right? So how is it that our immune system actually can tell that there is something going on, like an infection? A big important step in how cells of our body communicate with the immune cells in our body is the cells in our body always present different types of molecular signals to other cells. And these molecular signals are meant to sort of provide a glimpse of what's happening inside of that cell in your body. Right? This is very important because many of the immune cells, things like lymphocytes, macrophages, neutrophils that we looked at on the previous slide, right? I mean, they are cells. So that means if they go ahead and come across a cell in your body that's infected, right, they have to somehow be able to look into that cell and actually see that there's something going on. Now, obviously, they cannot do that because it's not like they're actually going to go into each cell and actually poke around, right? So that means they depend upon the target cell to actually advertise what is going on inside of it so that the immune cell that bumps into it has a chance to kind of check it out. And if everything looks normal, right, then that immune cell can move on and maybe bump into another cell. But if things don't look normal, right, then that immune cell might do something to that target cell. It might stimulate it to undergo apoptosis, for example. Okay. So here in the cartoon on the left, we have an infected host cell, and what it's doing, it is presenting bits and pieces of all kinds of proteins that are found inside of it to display its contents to immune cells that bump into it. So these little orange triangles represent key proteins that came from the virus particle, right? Maybe the protein capsule of the virus particle. Once it's inside this cell, it's infected, right? Bits and pieces of those viral capsule proteins can be presented by the infected host cell on its exterior. So those red triangles then would represent antigenic signals that came from the virus. Right? So how does the infected host cell do this? It does this by presenting small little peptide fragments, small little protein fragments, on special platforms on its surface. These platforms are themselves proteins made by the cell. And these proteins are called major histocompatibility complex proteins. So if we go over here on the right, so MHC, right, is major histocompatibility complex. So that refers to a whole set of genes that are located on a particular chromosome in our genome that form a complex of different genes to choose from. And the expression of these different genes make different proteins that can assemble as little platforms that then get transported to the surface of the cell. And on these platforms, they can actually carry bits and pieces of protein to go ahead and display on the outside. So in this cartoon on the right, the MHC proteins are these yellow kind of platformy looking things. Inside the cell, you have all kinds of proteins that are being turned over, recycled, whatever. Remember, the majority of these proteins are totally normal proteins that are found in this cell. So let's just pretend the brown protein, the blue one, the green one are normal everyday proteins. So when they are recycled, bits and pieces of them, represented by the dots, get displayed on the outside. So you can probably already figure out that if an immune cell comes along and bumps into this cell and just sees brown dot, blue dot, green dot being displayed on the outside, then the immune cell is probably trained to leave this cell alone. But in this case, in addition to that, we have protein that came from the virus, right? The orange protein that came from the virus. And so the little orange dot here is being displayed. So this implies that if an immune cell came along and bumped into this and saw this, that might activate the immune cell. So MHC are platforms that present peptides from within the cell to the immune system so that our immune cells can actually see, quote unquote, what's going on inside of the cell that they bumped into. There are two types of these MHC platforms. MHC1 is how infected cells present peptides that come from, let's say, a pathogen. Right? MHC2 presents perhaps the same type of pathogenic signal, but the difference is the MHC2 platforms are used by immune cells whose job it is to go around and maybe eat or take in examples of the pathogen without themselves being infected, and then processing these peptides and showing them off on the outside, right? So in other words, certain types of immune cells, and really only certain types of immune cells, use the MHC2 platforms because they are acting as a messenger right? They found something, right? They've processed it, and now they want to pass that message onto other immune cells. But because they're the messenger, they don't want to be the target of other immune cells actually wanting to kill them, right? So that's why presenting things on MHC2 prevents other immune cells that actually see and recognize this from killing the cell that's presenting it. Instead, it's a way to pass on or activate other immune cells so that they can then go off and find examples of cells that are actually infected with this signal. So almost all of the cells in our body then are able to present MHC1 on their surface in order to tell the immune system that they're in trouble or they're infected. Right? But only certain subtypes of immune cells also express MHC2 on their surface. So let's go back on the left here. So the infected cell, let's just say is a normal run-of-the-mill cell that got infected. Because it's infected, it presents the viral antigen on MHC1. Here comes a cytotoxic T cell, one of the lymphocytes, and it has a T cell receptor on its surface that is able to interact with the MHC1 and read that this is some sort of weird little peptide that the cell is showing the T cell, that the T cell has never been trained to accept as a normal peptide that's found in the body. Therefore, the cytotoxic T cell then will release certain types of cytotoxic messages directed at this infected host cell that cause the infected host cell to undergo apoptosis, right? So this infected cell dies as a way to prevent further release of more virus particles. So at the same time, if you go up to the top here, let us say that you have previously been sick by this same exact virus or a closely related virus, so that your immune memory, maybe months or years ago, already developed antibodies against it. So you have pre-existing antibodies in your blood, right? These antibodies will bind up the virus particles. By doing so, the antibodies help fight the virus in two ways. One is by binding them up like this over here. It makes it harder, or it blocks the virus particle from being able to interact with the surfaces of your host cells. So that's one way it prevents the spread of the infection. The other way is by coating the virus in this way. The stems of the antibodies that stick out can be grabbed onto by different immune cells like macrophages that make it much easier for the macrophages to go ahead and eat the virus particles. So in this case, the macrophage ingests the virus, it processes the virus, right? So the macrophage itself is not being infected, right? It's just doing its phagocytic job of eating the virus. And so it wants to present that same orange triangle on its surface to alert other immune cells. But again, right, it doesn't want to get killed by another immune cell. So it's presenting the red triangles on the MHC2 platforms, right, that only macrophages and certain other types of immune cells know how to present things with. So in this way, the macrophage then can talk to another type of T cell, in this case, a helper T cell. So this T cell has a different type of T cell receptor. You can see it's purple compared to the blue of the cytotoxic T cell, right? And this helper T cell then recognizes that there's this weird pathogenic signal that the macrophage is showing it, right? The helper T cell does not harm the macrophage. In fact, the helper T cell might send encouraging cytokines over to this macrophage that actually amplify the macrophage's ability to go out and eat more bacteria, right? But what the helper cell will do is it will now coordinate the activation of things like cytotoxic T cells, right? So this cytotoxic T cell might have just been floating around minding its own business, but then this helper T cell activates this cytotoxic T cell, so the cytotoxic T cell can now go around in the body looking for other cells to kill. And meanwhile, this helper T cell also activates our B cells, the B cells that have antibody or immunoglobulins on their surface, right? And it may stimulate these B cells to go ahead and become plasma cells to release even more of these high quality antibodies. Okay, so that's kind of like a whole semester of MCB461 crammed into one slide. But it's important for us to be able to remember this stuff as we talk about cancer. One of the early experiments that revealed that cancer and the development of cancer really has a lot to do with our immune system had to do with early observations of transplanting tumors in different strains of lab mice. So let's say you have two strains of lab mice, the blue one, strain 1, pink one, strain 2, and these represent two different genetic backgrounds, right? So maybe in your animal facility at your institution, you have a whole colony, several cages of strain 1 and several colonies or cages of strain 2, right? And they're different mice, but within each strain, they are genetically identical, right? So all the blue mice share the same genetic background. All the pink mice share the same genetic background. Right? If you expose these mice to carcinogens and you actually induce the formation of tumors in them, so let's say you develop these blue tumor cells, and you develop these pink tumor cells, right? And these tumor cells will have different types of, let's say, MHC on their surface, right, as a way to kind of communicate with the immune system. So if you then transplant some of these blue tumor cells into another blue mouse, you would not be surprised that a new tumor would start forming in that other blue mouse, right? Because after all, if this tumor formed in a blue mouse to start with and got to the point where it actually is like a cancerous tumor, right, then you might expect that it has already somehow found a way to avoid the immune system in the blue mouse. And because all blue mice have the same genetic background, right, there's a very good chance that you inject it into another blue mouse and that cancer will just go ahead and continue to grow. But if you take the blue cancer cells and inject them into a pink mouse, what the early researchers found was that the tumors don't grow, right? Why? Because the pink mouse has a different genetic background. That means its immune system, or the genes that make up its immune system, is different enough that whatever tricks the cancer pulled off in the blue mouse to avoid the blue mouse immune system, those tricks don't work in the pink mouse. And this side over here is the same exact thing, but you're doing it in the pink mouse, and you see that the pink tumor cells don't grow in the blue mice, but they grow in other pink mice, right? So these early experiments really showed that it has to do somehow with the genetic background, and they realized that it was most likely something to do with the immune system. Some other experiments building upon this, I think this is a very important one, it's a very classic one. If you take some mice and you go ahead and let's say you have a mouse that developed a tumor from this same mouse strain, right? And let's say you took some of the tumor cells from that other mouse and you killed the tumor cells, right? So that they cannot actually divide anymore, but the cells are actually still more or less intact, right? So that means they still have on their surface all kinds of antigens and proteins, right? That can be read by an immune system. So if you irradiate these, right, you hit these cells with radiation to kill them, and then you inject these killed tumor cells into this otherwise healthy mouse, right? What you find is that, well, obviously no tumor will grow because the tumor cells were dead, right? So what you do is you then wait some period of time and allow this mouse's immune system to have had an opportunity to see these different types of antigenic signals on these dead tumor cells that are actually no real threat, right? And what's surprising is that later on, if you go back and take some of those same tumor cells, but instead of killing them with radiation first, so these are now live tumor cells, and you inject those into the same mouse that got these dead tumor cells to start with, surprisingly, this mouse does not develop tumors. Why? Because this mouse had an opportunity for its immune system to see these different antigenic signals on the dead tumor cells and was able to develop immune memory against these antigenic signals, right? So that later on, weeks later, when it actually got a dose of live tumor cells, its immune system already had the memory to recognize very quickly that these things are a threat. And so they were able to kill these tumor cells and not let the tumor grow. In other words, right, you have vaccinated this mouse against this tumor type, right? This is the whole principle of how vaccines work. You expose the body to the antigenic signals that you want the immune system to have a chance to learn without actually exposing the body to the harmful live pathogen that carries those antigenic signals, right? Because obviously you expose yourself to a live pathogen. This is how Mother Nature works. You have to get sick from it and actually survive and beat it in order to gain the memory, right? But a vaccine is simply taking the antigens, whatever antigens are determined to be the most sort of immunoreactive, and giving those to the immune system so it can learn without actually the risk of getting sick. So just a follow-up here. If you were to take live tumor cells from a different tumor, right, one that was different than the one that this mouse was vaccinated against, if you inject those into this same vaccinated mouse, unfortunately, it will still develop the blue tumor, right? Because it was vaccinated against whatever antigenic signals are present on the red tumor, right? So I think this very clearly illustrates, again, this idea of how immune memory plays an important part. So I mentioned that not all tumor cells, right, would be considered equally immunogenic. In other words, not all tumor cells would present the same level of key antigens on their surface that would wake up or stimulate the immune system. Because after all, as we've been learning, all tumor cells are just completely messed up, jumbled, random mutations, right? So it'd be kind of naive to think that each type of tumor cell is equally kind of equally detected by the immune system. So some tumors are less immunogenic, meaning that they are less visible to our body's immune defenses. So it may seem counterintuitive, but if you think about it, a less immunogenic tumor would actually translate into a more aggressive tumor because it's able to kind of slide under the radar of our immune system, right, and continue growing without the immune system detecting it. So this is another classic experiment that illustrates this. Let's say you have a set of these dark brown wild type mice, and you have these light brown immunodeficient mice, right? So their immune systems are compromised somehow, so they don't work, their immune systems don't work right. You expose all of them to the same type of carcinogen, and so you develop different examples of tumors. Some of these tumors that develop in the brown mice would tend to be weakly immunogenic tumors. Right? Again, they don't really raise the alarm of the immune system because whatever mutations they have, right, they're not very immuno detectable. Right? So we say they're weakly immunogenic. They do a weak job of actually eliciting an immune response. So that's why they're allowed to grow. And so in the wild type mice, you would tend to only find these weakly immunogenic tumors. Why? Because the more strongly immunogenic tumors in these healthy mice would have been quickly identified and eliminated by their immune systems. On the other hand, in the immunodeficient mice that don't really have an immune system, you'd find a mix of both weakly immunogenic as well as strongly immunogenic, right? Because without a robust immune system, it almost doesn't matter if the tumor is weakly immunogenic or strongly immunogenic. It's just not properly dealt with. But when you go ahead and you take these tumors and you transplant them into other mice, what you would find is that these blue weakly immunogenic tumors can be transplanted and they will grow in other brown mice that have healthy immune systems, because that's what these were to start with anyhow, right? And not surprisingly, these same tumors would also form in these RAG2 deficient, immunodeficient mice, right? But if you just take these tumors over here, right, again, of the red, strong immunogenic and the blue, weak immunogenic tumors, you transplant them into mice, and only the blue, weakly immunogenic tumors end up forming in the healthy, strong brown mice, right? Whereas both types of tumors form in the immunodeficient mice. So this, I know this is a little confusing, but go back, talk your way through it, right? And keep in mind that a weakly immunogenic tumor actually means that it does a bad job of alerting the immune system to its presence. So therefore, it actually benefits from that and becomes a more aggressive tumor over time. Here are some clinical data. This is some very interesting clinical data that ties in the discussion of the immune system with cancer. So what we're looking at here are the rates of cancer of different types: breast cancer, prostate cancer, colon, ovary, trachea, in two groups of patients. Both groups of patients are patients who are immunosuppressed. Right? So among these are HIV/AIDS patients who became immunosuppressed because of the HIV infection leading to AIDS. And you also have a group of patients who are transplant patients, people who have received donated organs. And as part of their organ transplant surgery, they have to take strong immunosuppressive drugs to keep their body from rejecting the tissue that came from somebody else. So this first group of cancers here are cancers that are not associated with any kind of infectious agents. Remember, we said that viruses can be linked to about 1/5 of all different cancer types, right? So these over here are examples of cancers that are not linked to virus: breast, prostate, colon, ovary, lung cancer. Right? If you look at the incidence rates of these different types of cancers in these two groups of immunosuppressed patients, this is what we call an SIR, or standardized incidence ratio, comparing the rates sort of as a fraction compared to the normal population. You'd see that there's very little evidence of a tremendous increase in things like these cancers in these types of immunosuppressed patients. Right? Maybe the highest one would be maybe some examples of lung cancer in maybe HIV/AIDS patients. So maybe they have something like a 5 times higher risk, right? However, instead of these cancers, what if you looked at cancers that we know are linked to viral infection, right? Because then you might already think, oh yeah, therefore your immune system might have a role in this. So if you look at that same type of data, but looking at viral-related or pathogen-related cancers, like different types of lymphomas caused by Epstein-Barr virus, other types of cancers caused by things like Helicobacter pylori, so stomach cancer. When you look then in these cohorts of immunosuppressed patients, you will see, right, that these patients develop these types of cancers at a far, far higher rate compared to normal. So up here, when we said, oh, 5 times higher than normal, maybe some of you are like, 5 times, that sounds like a lot, right? But really, it's not, right? Because if you're saying something like, 5 in 1,000 people develop something, and 5 times higher is 25 out of 1,000, oh, okay, well, that's not that big of a deal, right? But when you look down here, with these pathogen or infection-related cancers, right, some of these you get on the order of 1,000+ fold higher incidence rates of these cancers in these immunosuppressed patients. So again, some more compelling evidence that having a robust, healthy immune system is important in detection of cancers, and in this case here, maybe in even preventing the start of certain types of cancers related to pathogen infection. So this is just a table here that is similar to the previous slide, but just shows it as a big table, talking about when you look at certain sites of cancer, like non-melanoma skin cancers, and you look at these are in immunosuppressed transplant patients, right? Maybe they observed 127 cases in these immunosuppressed patients compared to the number of expected cases. If they looked at the same number of people in the general population, right, then you see that these immunosuppressed patients have maybe like a 25-fold increased risk of developing skin cancer. So take a look at these, just as a way to appreciate then and really get home this or bring home this idea of the immune system being very important. So our immune system also, we talked about how cells like macrophages are often attracted to a tumor that is getting ready to bust out of its in situ containment, break free from the basement membrane, spread through the surrounding stroma. And we said that macrophages are oftentimes attracted there as part of the wound healing response. And these macrophages end up helping the tumor cells escape, right? So other tumor cells might also be attracted, but for reasons that are more beneficial to us fighting the cancer. So one example are certain lymphocytes. When you take a biopsy of a patient's tumor, oftentimes they will look for any evidence as to whether there is an infiltration of lymphocytes coming to help fight and destroy the cancer cells. So this image in the middle is particularly useful. So this is a section of some kind of a tumor where the majority of the stained cells here are just tumor cells. But when you actually take certain types of immunotherapy drugs or certain types of chemotherapy, I should say, they can stimulate the immune system's awareness of the cancer. And so paclitaxel is a very popular anti-tumor drug. And you can see now, after the patient is undergoing treatment with these chemotherapies, you have a lot of lymphocytes that have now come into the tumor and are paying attention and actually fighting to destroy the tumor cells. Here's a Kaplan-Meier plot on the bottom that shows that patients who, when they first have a biopsy done, if their tumors already show evidence of many of these tumor-infiltrating lymphocytes in them, that would be the blue patients, their survival is much better than the patients whose tumor biopsies showed little evidence of these lymphocytes attacking the tumor. Right? So I mentioned that cells in our body, right, they're always displaying different types of signals that represent what's happening inside of them. You can imagine then that different types of cells in our body, because they use different proteins, would have different examples of proteins shown on their exterior. Some of these signals then can be used as antigens and recognized by our immune system as evidence that a particular cell has gone a little strange or weird and is showing off things that it shouldn't normally be. So these could be, for example, tumor-specific antigens, or TSAs. So these would be proteins that are uniquely expressed by a cancer cell. So there are some genes, for example, that are normally not turned on in a healthy cell that might be switched on in response to different stresses, like cancer. So then suddenly these evidence of these little peptides would appear on the outside of the cell, right? So they would be unique to this cancer cell. Other types of antigenic signals might be tumor-associated antigens, right? So in other words, normal proteins that maybe are expressed by different cell types normally, but a particular cell type normally wouldn't be expressing this on its surface, so it's out of place. Here are some examples of these TAAs, or tumor-associated antigens. Here, for example, is an enzyme, a tyrosinase antigen, that's being stained for in melanocytes. So here you see the melanocytes. They're hanging out with the keratinocytes in our healthy epithelial layer of our skin. Right? So here's our dried dermal layer, epidermal layer right here. Here are melanocytes. Right? And so these melanocytes express this particular tyrosinase, whereas the keratinocytes normally don't. Here's another one. This is in the developing sperm cells in the testes. Developing sperm cells present this enzyme known as MAGE1. And you can see they're pretty well stained here, right? So that these spermatogonia express MAGE1, but other types of cells shouldn't. So that means if somewhere else in the body you encountered cells that were expressing, for example, MAGE1, that might be evidence that the signaling pathways inside of the cell have gotten messed up, right? Because really only the developing sperm cells should be expressing them. And so our immune system can pick up on that. So here are some examples of these tumor-associated and tumor-specific antigens. Again, just to give you some ideas of how not only the presence or absence of these antigens can be used as a signal that something is wrong with the cell, but also varying amounts of these. Things that are normally expressed in moderate amounts, if suddenly they're expressed in huge amounts, that might also be evidence that the cell is getting messed up, right? And the immune system can pick up on that. So lastly, I wanted to talk about one of the very clever ways that cancers can avoid being detected. We've spent much of this lecture talking about how cells have to present things on their outside, on their MHC protein platforms, in order for immune cells to be able to take a look. So you can imagine then that these MHC platforms are proteins themselves, and they're coded for by genes. So certain mutations that dramatically mess up the proper expression and formation of these platforms might have a big impact on the ability of the cell to properly display its contents. And so certainly in the course of cancer cells stumbling upon different kinds of random mutations, stumbling upon a mutation that shuts off the expression of these MHC platforms is a really good way for the cancer cell to just avoid being detected by the immune system. So on the left here is an example of a colorectal tumor. Here, that's what this mass is on the upper part of the cell. The lower part is still normal, healthy epithelium, so various ducts and things over here. And what they're staining for is the presence of those MHC proteins on the surfaces of the cells, right? So the normal healthy cells express a lot of MHC because they are doing the right thing by showing off their contents to immune cells that bump into them. Whereas you can see the tumor cells have stumbled upon the mutations to suppress MHC. So therefore, these cancer cells are not displaying on their surface evidence of what's happening inside of them to the immune system. And then this last slide, sorry, we're a little bit going over here, is a nice way of giving you some other examples of different types of mechanisms that cancer cells employ to be able to evade or hide from the immune system. Okay, so some of these we will talk about in the next lecture. So really, I just wanted to make sure you had a copy of this so you could use it when you're reviewing.